Hello all, welcome to Microbiology Central. So today's lecture is going to be on the topic Bacterial Taxonomy. Bacterial taxonomy is a classification of bacteria into ordered groups based on various approaches. There are three main approaches: phylogenetic, Adansonian, and genetic. First is the phylogenetic classification. Phylogenetic classification groups bacteria that are related on evolutionary basis. They are the measure of genetic divergence of different biologic divisions. There are different ranks designated for classification of a bacteria based on the phylogenetic analysis. They are kingdom, division, class, order, family, genus, and species. This is the hierarchy of biological classification. First comes the kingdom, and then phylum, class, order, family, genus, and species. For example, the microorganism Salmonella typhi belongs to the genus Salmonella and species Typhi. Burgess' Manual of Systematic Bacteriology is a manual which is a compilation of names and descriptions of newly isolated bacterial types. Burgess' Manual of Determinative Bacteriology serves as an aid in the identification of bacteria that have been described and cultured. For identification and classification of bacteria, important characters like morphology, staining, cultural characteristics, biochemical reactions, antigenic structure, guanine-cytosine ratio are considered for various members to be distinguished. Next is the Adansonian Classification. Adansonian classification was proposed by Michael Adanson in the 18th century. In this classification, the characteristics of the bacteria is taken into account and they are grouped on the basis of similarities in large number of characters. Comparison of very large properties of several organisms can be done at the same time using different bioinformatics tools. This analysis helps in obtaining the list of strains based on degree of similarity in their properties. Thus, several broad subgroups of bacterial strains can be identified which are further divided into species. This classification is also called as numerical classification or computer taxonomy or taxometrics. Next is the genetic classification. Genetic classification is based on the degree of genetic relatedness of different organisms. This classification is the most fundamental since all properties of bacteria are regulated by their genes. The genetic material DNA is extracted from a pure bacterial culture and the GC content is estimated. The nucleotide base composition and the base ratio, that is, the adenine guanine is to thymine cytosine ratio, varies among different organisms but are similar in the same or closely related species. DNA sequencing has become a routine laboratory procedure, and the comparison of DNA sequences of divergent genes can give a measure of genetic relatedness among those species. Hybridization of DNA to high-density oligonucleotide arrays are used for species identification. Next is the intraspecies classification. Intraspecies classification of bacteria is often necessary for diagnostic or epidemiological purposes. They are subclassified on the basis of biochemical properties, antigenic structures, bacteriophage susceptibility, or production of bacteriocins. A species may be divided first into groups and then into types. The application of newer molecular techniques has led to discrimination in intraspecies typing. Molecular techniques are of two types: phenotypic and genotypic. Phenotypic method includes electrophoretic typing of bacterial protein and immunoblotting. Genotypic methods include Southern blotting, PCR, and nucleotide sequence analysis. Thank you.